So hello viewers, in this video you're looking at phenomenon called quorum sensing. So what is quorum sensing? This is how bacteria communicate with each other. So this is a cell-to-cell communication. So with the help of this particular communication, processes like plasmid transfer, conjugation, virulence, motility, antibiotic production, etc., and many other processes are regulated with the help of this particular communication. So sporulation, so how do they communicate with each other? They have a special signaling molecule. It produces a special inducing molecule called autoinducers, which in turn regulate gene transcription and how the gene expresses. So quorum sensing is a regulation of gene expression in response to population density. So when the population density is high, autoinducers are produced in high number, and which induces the expression and production of a large amount of autoinducers themselves. So thereby, as per population density, induces a transcription of genes. So this quorum sensing phenomenon is exhibited by both gram-negative bacteria and gram-positive bacteria. So when in case of gram-positive bacteria, it is with the help of compounds like oligopeptides, and while in gram-negative bacteria is by AHLs or N-acetyl L-homoserine lactone. In gram-positive bacteria, these oligopeptides cannot penetrate into the cellular membrane, so it needs a sensor to sense these oligopeptides in response, a response regulator which needs phosphorylation so that the genes are activated and the gene expression takes place, exhibiting quorum sensing. So when you take a gram-negative bacteria which produces AHL, being a lipophilic molecule, it can penetrate in the cell membrane, cellular membrane. It has the primary gene responsible for the expression of AHL, positive expression of AHLs is the LAX-I gene, which is activated by the LAXOR. So only when it binds with the AHL, the LAXOR activator gets activated, producing a lot of AHLs, increasing number of AHLs. When the increasing number of AHLs are produced more than the threshold level, transcription takes place in response to increasing cellular cell density. So when the LAX-I gene is expressed at the base level, no luminescence is formed in bacteria like Vibrio fisher. So when this particular Lexor get activated in response to stimulation of AHLs. It produces the Vibrio fishery. It produces luminescence, blue-green light, in response to a large number of cell density, thereby producing an increasing number of AHLs more than the threshold level, thereby positive, the Lexor acts as a positive autoregulator, thereby generating increased transcription and in response to cellular density. So this is how quorum sensing happens in bacteria. So you can see in the next video for further. Thank you.